All right, so this presentation is going to be on the people eating plastic monster. It's a fictional story that revolves around the use of PETase, a PET degrading protein, and how its mechanism and kinetics work, and maybe how we can defeat this plastic monster. Okay, let's get started. First, setting up like the origin story. This is like there's like a big ocean of plastics, microplastics, and get struck by a lightning bolt. Yeah, there's like some static electricity, whatever, and it pulls all of these plastics together. We have the formation of an evil monster that eats people. This is the monster of in question. All right. So what do we know about this monster? Um, First, that it's made out of primarily PET. This is polyethylene terephthalate. This is a very commonly used plastic. It exists in a very long polymer chain. And you can see these little PET repeating units down here at the bottom. Um, all right. And then this is in a lot of different things like plastic bottles and clothing items. Um, and so the next question that we have is like, how does this monster exist, right? How does it keep its form all for reasons. First, there's like hydrophobic interactions. We can see from this image that there's like these benzene rings are very non-polar, and so they're going to attract other non-polar, right? We have these hydrophobic interactions here. Secondly, we have these van der Waals interactions because they're long, long chains that are very continuous and ordered, meaning that they can pack together very nicely and form a crystalline structure. This is really important for holding shape. This is analogous really to like lipid structures. The third reason is there's dipole-dipole interactions because there are like electronegative differences, especially along the carbonyl carbon, where you have like the negative and the positive charge, those by interact. Lastly, we pi stacking. I have an image here of the two pi rings or benzene rings that overlap, and you have these interactions here. We'll talk a little bit more about like wobbling later, but those are basically the mechanisms. All right. So this is where our unsuspecting Jera steps in. This is Ideonella sacchariensis. We're just going to call it the bacteria for now. Basically, it was discovered in Kyoto Institute of Technology in 2016, very recently, in sediment samples outside of a recycling factory. Right, they noticed that there was like PET. They were just sampling for like plastics in the soil. They found PED like byproducts, right, of degradation. And they noticed there's a lot of them, more than like natural cycles. And they discovered that there's a bacteria that was using PETase to break down PETs. And it has a really interesting ancestor that they discovered through bioinformatics work and like homologous sequences that is used, in nature to break down like waxy substances on plants, right? The mutation frequency shows that it's probably like 15 years ago that this evolved, a little bit after PET became very popular. All right, so what does this molecule look like? Three ways that we're going to talk about it. First, on like the primary structure, it is about 31 kilodaltons, so kind of a small an enzyme, and it has only 265 amino acids. Additionally, there's only one single chain and with no subunits. So that's also the quaternary structure. On the secondary structure, there we has both alpha and beta helixes. We have a little description here. And lastly, on tertiary structure, there's two disulfide bonds, which is one more than normal. We see with like cutanase. And there's also a number of hydrophobic regions, which is really important for the active site, especially considering that like the benzene ring is nonpolar also. It wants to have a nonpolar active site. All right. So how does it function? Uh, two two big steps. First, there's acetylation, then deacetylation, right? This is the first step pulls off the ethylene glycol, the second step pulls off the terephthalic acid. Both steps involve a nucleophilic attack and then a tetrahedral immediate or intermediate. All right, so this is basically the structure, except I don't have under here, under reactants, there's going to be the ES free energy dip that we'd expect for when it like attaches. All right, so here is the enzyme. We have the substrate, or sorry, the substrate, and this is the enzyme active site. We have an oxy anion hole, which helps stabilize the polymer. We'll describe that a little bit later. And this is the catalytic triad. We have serine, histidine, and aspartate. All right, so this is where it just fits into that active site. four ways that like it makes it that it speeds up this process. First, there's wobbling. I have a little image here of the tryptophan residues that like wobble around. This gives them like a degree of freedom for grabbing onto that polymer and holding it into the active site. This is really important for keeping that stable there to ascend into like that transition state. All right. Um, so and this is also another idea of like uh, stacking that I talked about earlier. All right. There's also nonpolar hydrophobic interactions with this tryptophan interaction. All right. Thirdly, there's going to be nucleophilic attack. This is this image here where this is how it gets into that transition state. The serine attacks the carbon here attached to the carbonyl. And then on stabilizing this interaction here, we have the oxyanion hole that like creates hydrogen bonding with the oxygen here to make sure that this attack actually works and is stabilized. All right. And so this is the intermediate state that that forms. That's that like dip in that little curve there. Um, so here's the tetraoyl intermediate. This allows the popping off of this ethylene glycol here by stabilizing the transition of oxygens to that of serine. All right. Um, After that, it just pops off, and we have the intermediate number two. Um, and yeah, ethylene glycol is released. This is the deacetylation process, that second step. This is the third intermediate where we have a nucleophilic attack by water, and this water is stabilized by the catalytic triad, here the histidine specifically. And also, again, the oxyanion hole stabilizes that oxygen there through hydrogen bonding, which is really important. All right, now at the end we have the enzyme in the product. The enzyme is back to normal, the freed up active site, and then the terephthalic acid and ethylene glycol have both been released. This is how the process ends and restarts. All right, let's talk about the kinetics. Two parts of this. First is the Km value, which is, as we learned, is the concentration of substrate necessary to get to half velocity. As you can see, it's 4.6. This is comparatively very high in relation to all these single point mutations that speed up the rate of reaction, right? This means that more substrate is needed, meaning that it's less efficient of a process. Secondly, on catalytic efficiency, we can see that it's very low in comparison to all of these other measures of single point mutations. So this basically shows you that PET degradation is really slow, right, and inefficient. So there's four big reasons for this. Firstly, this is like highly crystalline structure, which just kind of makes it inaccessible to these like nucleophilic attacks and getting into that active zone. All of that just makes it a little harder, and therefore a little slower. We can kind of think of this analogously to like cellulose degradation in these like long polymer chains just being inaccessible. All right. Secondly, PET is insoluble solid, meaning that there's just like lower interaction frequencies and also like lower diffusion rates from the active site, meaning that just things proceed a little slower. It's nice that the PETase molecule is a little smaller, which means it has a higher degree of freedom moving around higher interaction rates, but it just still, PET being solid is a problem. Thirdly, it's very non-functional. The enzyme itself is not thermostable at high temperatures, and PET is very inflexible and hard to degrade at those temperatures where PET is most accessible, right? Or where PET is or PTase is most functional. So you have like this like issue there. And fourthly, PET is just naturally very stable. This is why it's used in packaging, especially like for food and stuff like that. It's very inert and resistant to reactions, and that is a problem. So how do we make sure that we can efficiently address this monster by like making the PETase more functional? Two ways we can do this. First, this page here is talking about protein engineering, which is essentially single point mutations that we can do to the protein itself to make it more efficient. There's a number of these that we've discovered since 2016 that makes us more efficient. One of these really important ones is where is it? S29 or 269T, which increases activity up to 400%. This is like a not actually in the active zone, which is actually kind of important. It's actually like close to it and just kind of helps everything function a little bit better. I linked the article in my sources also. But yeah, all of these were discovered through machine learning, which essentially means that we don't understand why it makes it more efficient, but we just understand that it does. We can make crystallography testing to understand why the structural changes, what that looks like, and why that might be important. But these are all been tested, and we figure out, that these are the experimental results here. Obviously, fast PETase is very, very efficient at low temperatures. All right. The second way we do this is genetic engineering, which essentially means we can increase the number of these proteins within the cell that are being excreted to address the PETase monster or PET monster. First, we intuitively like we need to use protein or genetic engineering to get these protein engineered proteins into the cell to like make the cell more efficient. And this is used like CRISPR insertion, but we can also increase the copy number of the like increase the expression of it within the protein or within the cell. First, you can like upregulate the expression of the protein by like changing the promoter. We have the promoter here and the operon that it controls. We can increase the rate of expression here by using like something like T7 or TAC interesting, like the byproduct of P degradation, which is like ethylene glycol, is used in stimulating the expression of PTase, which is kind of interesting. All right. And then secondly, we can do increased copy number, which essentially means that you just create like plasmids, insert them into the cells, and make more locations for that transcription to happen, more mRNA with more proteins. A little bit less reliable, but alternatively like another option. All right, so the plan essentially is, while I'm wrapping up, we take, we bioengineer them, and then we incubate and PET agar, which we'll kind of select for ones that are more efficient and that can survive on their own. And then as we allow the cell density to grow, we can do certain tests to see which ones are more efficient, et cetera, et cetera. And then at the end, we can take this like dense mass of cells, dilute it, and then use it to inoculate the monster with this like super bug, right? And this is what that would look like. You just spray it on the monster. He's like coming at us. We just and he's spraying the bugs on him. Okay. And then the bugs would just start eating him, right? Because they're so attuned to the PETA, so that the PET that he's made out of, it just starts breaking down and he starts melting, right? Those bonds start to dissolve, and it just becomes a soup of ethylene glycol and terephthalic acid. And so at the end of the day, the monster's been defeated, and it's a happy ending. So I hope this was informative and good use of your time. So yeah, thanks.